So, first of all, good morning, and it may not come as a surprise to you, but I'm very grateful to the organizers to be invited to the conference. So, to this nice venue, the nice conference. Many thanks for that. The work I'm going to present today is was mainly done. Which one's the pointer here? Okay. So mainly done in Lübeck in the pathology department, which is in the very north of Germany. And the other part, or it was finished up, this is at the Helmholtz Center in Munich. This is the Hämatologicum of the Helmholtz Center. And there we are mainly interested in T cells and in the development of autoimmunity and the regulation, post-transcriptional regulation of mRNAs. So my talk is divided in 4 parts, and the first part is about microRNAs, microRNA profiling, and diffuse large B-cell lymphomas. So this is where the B-cells come into play. And then we've learned some lessons from that, and then I will go on and talk about expression of microRNAs and microdissected samples and expression of microRNAs in formalin-fixed, paraffin-embedded tissues, and also microdissection of that, and measurement of microRNA expression in FACS-sorted samples. So first of all, let me introduce you the microRNA. So Michael Pfaffel has done that already a little bit, but I want to go through it anyway, because I want to drive home one point, and that is, so you've got the primary microRNAs, which are expressed as longer precursors, about several thousand base pairs long or so, and the primary microRNAs, they are processed by a microprocessor complex, which is shown here, composed of DROSHA and DGCR8. The pre-microRNA, which is now about, say, 90 base pairs long, is exported from the nucleus to the cytoplasm, and in the cytoplasm is recognized by the endonuclease dicer. And dicer does one thing: it removes the loop of the stem loop, and now we have a mature double-stranded microRNA, and just one strand of this mature microRNA is loaded onto the RISC complex, the RNA-induced silencing complex. And now the RISC complex, including the microRNAs, searching for a complementary sequence, and it mostly finds it in the 3' UTR of transcripts, so the untranslated region of a transcript. And now 2 things usually happen. The first thing is a block of translation, so there's no protein produced anymore. And the second step is deadenylation and degradation of the RNA. Now, this process seems like, if I describe it as such, like if you kick it off and then it goes through, and eventually you will have a mature microRNA, and there are no steps in between. But don't be fooled. Every single step can be regulated in this process. And this is really important, because if you do microRNA profiling, you really want to profile the mature microRNA, which is biologically relevant, and none of the others. And in embryonic stem cells, for example, there's one microRNA, the LED7 microRNA family, which at the stage of the pre-microRNA here is oligouridylated. That means there are certain enzymes that add uridines to the 3' end, and once there are 5 or so uridines at the 3' end, this pre-microRNA is not processed anymore to become a mature microRNA, and this has got an important impact on the embryonic stem cells. As long as there are no mature, let's have microRNAs in embryonic stem cells, the stem cells keep proliferating and do not start differentiating into other cells. So it's really important to measure the mature microRNA. And the first microRNA profiling was reported in 2005 by Lu and colleagues, and they have used about 300 different normal and tissue samples and measured about 200 microRNAs. And they did that using certain very specific FACS method, which I think has never after been reported. But then about half a year later or so, the Life Technologies had a specific kit out to profile 157 microRNAs based on the qPCR method. And because the microRNAs are only 22 nucleotides long or so, it's not very easy to go through a qPCR process. So you need a specific stem loop primer here at the beginning and perform first strand synthesis for each individual microRNA. And once you have done that, the first strand is elongated, and then you can use a specific forward primer and a ubiquitous reverse primer, and then you can amplify the microRNA. And this is specific for the mature microRNA. Now there are other specific pitfalls in the measurement of microRNAs. One pitfall is that due to the short size, the microRNAs are very similar to each other, and they sometimes only differ by one nucleotide or so, which you can see here in the case of the LED7A and the LED7C microRNA. So if you use hybridization-based methods, you will not only detect the LED7A, but also at the same amount LED7C. So this problem can also be circumvented by using the qPCR-based method, which I just introduced, and here you can see the result of that. So this is not our work, but it's sort of published by Life Technologies. So the LED7A detects LED7A 100% and LED7C only 3.7%. And then there's another difference to maybe normal mRNA measurements, and that is that microRNAs can be expressed at a very high level in individual cells. So let me just give you one example. The microRNA 122 is highly expressed in liver cells, and there can be more than 50,000 molecules of this one microRNA in one liver cell. So as Michael also pointed out, you need a very high dynamic range to measure the microRNAs that are of biological relevance. So now let's come to the diffuse large B-cell lymphomas and start with the normal tissue, the tonsils. So this is the tonsil here. If you open up your mouth, you can see it. And within the tonsil, there are the airways, and they are lined with epithelial cells, and there are germinal centers. And in the germinal centers, the B cells develop to become plasma cells or memory B cells. And when something goes wrong during this development, then diffuse large B-cell lymphomas develop. They can be of the activated B-cell type, of the germinal center type, and this becomes then a very aggressive lymphoma, and it can be treated rather well, I must say. You just use ordinary immunochemotherapy, and then about 50% of the patients can be cured, but still there's the other half. And we know that microRNAs are involved in the differentiation of cells and in the proliferation and in apoptosis. And tumor cells are sort of defected or not different in proliferation, differentiation, and apoptosis. So we thought, and others also, of course, that it's a good idea to do microRNA profiling on diffuse large B-cell lymphomas and other tumor tissues. So that's what we did. Here you can see a heat map of 57, 58 diffuse large B-cell lymphomas and 7 lymph nodes. And you see that only 4 microRNAs are upregulated and all the others are downregulated in diffuse large B-cell lymphomas. And this makes sense because if there's dedifferentiation and microRNAs are involved in differentiation, then you would expect sort of that in tumors most of the microRNAs are downregulated. And this was published already in 2008, and I must say I'm kind of proud that these 4 microRNAs have sort of passed the test of time. There have been other studies now, and these microRNAs have always been shown to be involved in overexpressed in diffuse large B-cell lymphomas, and the microRNA 210 can even be detected in the blood serum of DLBCL patients. Now, I told you about this published story. What I didn't tell you about is the one story within this story that we discovered that it's very important to look at very pure samples, as we have heard many times now. as well. And that is, if you look at the microRNA 205, you see and compare the expression to tonsils, which is shown here, that in most of the diffuse large B-cell lymphomas, it seems to be absolutely depleted of the microRNA 205. And we were getting really excited. We thought we find kind of a marker or maybe marker genes that are regulated by this microRNAs that are specific for diffuse large B-cell lymphomas. And at that stage, we wanted to have a closer look in tonsils, which are the cells that actually express the microRNA 205. And we did that, and we wanted to use laser capture microdissection. We had no experience doing that. And so we did one first thing. We just took ordinary tissue culture cells and looked at the expression of one microRNA that we knew is strongly expressed in these cells, which is the microRNA 214. In this case, we took 100 cells, and these 100 cells are in about 5 microliters that are necessary to perform the qPCR that I introduced at the beginning. Then if you've got these 5 microliters, you do no purification of the RNA. You just cook the cells for 5 minutes at 95 degrees. And then the whole content, 100% of the content, is within the 5 microliters that you actually want to analyze, and you do then the first strand transcription, and after that the normal qPCR reaction. And it turns out the results that you get are what you actually would expect. So the 100 cells that I counted here, the CP value was set to 100%, and then I get the results for the respective dilutions. And it turns out that one cell, the microRNA content of one cell can be easily detected. And why wouldn't it? Because there are many copies of this microRNA 214 within this one cell. So we know we can detect microRNAs in single cells or in very few cells. Then the other thing that, sorry. The other thing that we did is we used laser capture microdissection, and I used 3 different microRNAs that I knew were highly expressed, the 122 in the liver cells, the 150 in the spleen, and the microRNA 1 in heart and muscle tissues. And we took 25 cells and we microdissected some of them. And as expected, we find strong expression of microRNA 122 in the liver, 150 in the spleen, and the 1 in the heart. So that encouraged us to go on and dissect the individual tissues of the tonsil. And now these are not single cells, and they were purified, and we produced first strands and measured cells from the germinal center as shown here, marginal zone B cells and epithelial cells, and this is a logarithmic scale. And to our very large disappointment, we found that the microRNA 205 is not expressed in B cells or even hematopoietic tissues, but is expressed in epithelial cells. So at this point, I have to state that we use diffuse large B-cell lymphomas or tissue from the diffuse large B-cell lymphomas that is more than 90 or at least 90% pure diffuse large B-cell lymphomas. And when we then went back to have a look at these tissues here, we realized that all the microRNA 205 measurements that we got from that were actually due to epithelial cells and not to the tumor cells. So that we learned the hard way. And to add some value to this finding, we were thinking, we wanted to see whether the microRNA is also expressed in carcinoma cells, so tumor cells that are derived from epithelial cells. And we found, yes, it's also highly expressed. And we then looked at dilutions from Ku 1919 carcinoma cell lines, and these cells are diluted in cells from another cell line, which does not express the microRNA 205, and we find that we can detect one microRNA 200 or the microRNA 205 in a mixture of 100,000 other cells. So how is that important? We heard already yesterday that if you look for metastasized cells, you go to the sentinel lymph node, and then the pathologist makes sections, and immunohistochemistry either shows you or shows you not the metastasized cells. And this is a problem because the whole lymph node is shaped like a soccer ball, and if you make a section from that, you either find the cell as a pathologist or you don't find it. In the case of carcinoma cells in a sentinel lymph node, you can take the whole lymph node and you just grind it, make total RNA from it, and measure the microRNA 205, and it's easy to detect that, and this is shown here. And importantly, the microRNA 205 is not detected in lymph nodes. So, as I stated at the beginning, I was working in a pathology department. In a pathology department, you usually have dungeons full of valuable formalin-fixed, paraffin-embedded tissues from humans. And this tissue is usually not easily accessible to profiling on mRNAs. But our question was, is it possible to profile these microRNAs in these tissues? And we performed one sort of dedicated approach. We took 4 samples of liver cells and 11 samples of tonsil liver tissues and 11 tissues from the tonsil, and each time we divided that and formalin fixed half of the sample and the other half was cryoconserved, and the same was done for the tonsil samples. We then extracted the total RNA that was for the FFPE fixation that was done using an Ambion kit called Recover All. And then we performed microRNA profiling, so we made whole profiles from each of the samples. And you can see here that for most of the samples, the microRNA profile stays the same. So no matter whether the microRNA is derived from the tonsils or from the liver, no matter if they have been cryopreserved or formalin fixed, the overall profile is very similar. But you will also notice that in the formalin fixed samples, the light shades, the light blue and the light green, they are a little bit higher, the CP values. So probably the microRNAs are not degraded during the process of formalin fixation in general, and this may be due to the small size, because the crosslinks that are introduced are usually introduced sort of every 100 to 200 base pairs, and microRNAs are just smaller and they probably don't get hit so often. So if you now look at microRNAs that are expressed specifically in these cells, like I told you already, the 122 is expressed strongly in liver cells, 194 as well. You can see that there's a huge biological difference if you compare the liver versus the tonsils. And the same is true the other way around. If you compare the tonsils against the liver cells, you find that the microRNA 150 is strongly expressed in the tonsil, but not in the liver. So the biological variance that you find is generally higher than the technical error that you introduce by formalin fixation. And now this is a table I just introduced to show you that the average CT of the whole profile of 157 microRNAs is about 1 to 1.5 CTs higher. So one other thing that needs to be mentioned is, and Michael also said that already, is that the microRNAs may be protected by the proteins around it, and also this may lead to the fact that the microRNA is not degraded during the treatment. You can now again do laser capture microdissection of these tissues, and you don't need to purify the few cells because there's not much formalin in these individual cells, and you just cook them, and you measure the microRNAs, and you can also measure microRNAs from small sample sizes. Now, one last point, you can also do the same sort of thing doing FACS sorting, and what I've done here is you just take some cells from the lymphocytic gate, like 10, 20, 50, 75, and 100 cells, sort them into a 96-well plate. Within this 96-well plates, there's already like 5 microliters of PBS. You shoot the cells into the 96-well plates, you cook them, and you perform first-strand synthesis and measure the qPCRs. It's really easy to do, and it's also easy to detect highly abundant microRNAs, which is shown here. Medium abundant are also kind of easy to detect, and low abundance, you probably need about 100 cells. So was kind of expected that this works from us at this point. But we decided to ask one question that we didn't know the answer in advance. At that point of time, there was a publication out that says that microRNA 155, which is an oncogenic microRNA, is overexpressed during the activation of T cells. And also there was a microRNA 155 knockout mouse, and this knockout mouse has got problems to produce antibodies. So we thought that maybe the TFH cells, or the follicular helper T cells, may not be able to provide help to activate B cells, and therefore we speculated that during the normal process of T cell activation in TFH cells, the microRNA 155 must be overexpressed. So we immunized mice using sheep red blood cells, and then waited for a couple of weeks, and then took out the spleen and isolated the CD4 T cells and stained the CD4 T cells for CXCR5 and PD-1, which are markers for follicular helper T cells. And we then microdissected these, FACS sorted these CXCR4, 5, PD-1 positive cells, and we compared them to CD4 positive cells. And as expected, we find that there's an overexpression of microRNA 155 in these cells. So we set up these FACS-based methods to analyze an ARI1 knockout mouse that my boss brought from Harvard. And this ARI1 knockout mouse, ARI1 stands for enhancer of RNAi. And in a genomic screen, it was shown that the ARI1 protein degrades siRNAs in C. elegans. And it was also known that RNAi doesn't work very well in CD4 T cells. So we thought that maybe this is a molecule that inhibits RNAi, and we wanted to investigate that further. So, but after setting all this up, we found that it's really, really hard to find an effect of ARI1 on microRNAs and siRNAs. And then science for me took a sharp turn, and ARI1 was discovered a second time, and as one of very few molecules that are bound to histone stem loops. So histone mRNAs are very different from all other RNAs because they don't have a poly-A tail. They have a stem loop to protect them from degradation. And the stem loop binding protein is bound to the stem loop and ARI1, and it was not known why this is the case. So we have had this ARI1 knockout mice, so it was relatively easy to just start and have a look if ARI1 plays a role in histone mRNA degradation. And actually, yes, it does. And this effect was very easy to detect, so we kept on working on that one. And as you can see here, we used a construct that is an EGFP and a histone stem loop. And we performed in vitro transcription, and this stem loop here has then one uridine, so mimicking uridylation, up to 14 uridines. And when you shoot this construct into cells, you find GFP in the FACS in the cells that don't have any use, and you find very little GFP after you add 5 U's to this histone mRNA. So oligouridylation inhibits translation. And I then looked into that in much more detail, and I produced a qPCR assay that can detect this oligouridylated histone mRNAs, and I found that in ARI knockout cells over the whole cell cycle, in T cells here, the oligouridylated histone mRNAs are strongly increased. And so this is the end of my talk, basically, but you have noticed at the beginning the LAT7 microRNAs are oligouridylated, and there are now publications coming out where you find that piRNAs are oligouridylated, siRNAs, introns are oligouridylated. All the mRNAs that have adenines at the poly A tail, the end of the poly A tail quite often is also oligouridylated, and it is not quite sure at the moment why this is the case, what's the function of this oligouridylations. For microRNAs, sometimes there's just one uridine at the end of the microRNA, and that has been shown to help processing by dicer. But if there are several uridines, 2, 4, 5 uridines, then this inhibits degradation. So quite clearly there's a level of regulation that has not been appreciated so far, and which will lead to many further publications in the future. So this is the end of my talk, and these are the people who have contributed, and I'm happy to take questions.
Please, talk is open for discussions. Maybe I start first. So the amount of uridylation is connected to protection, meaning?
No. Normally for histone RNAs and microRNAs, as soon as there's what is called oligouridylation, not monouridylation, oligouridylation is usually a marker for degradation.
Okay. So can I assume if I analyze my uridylation in microRNAs that I can have a hint or a marker for stability?
Yes. I think uridylation for microRNAs, well, again, there's a difference. Mature microRNAs that are oligouridylated are degraded, but oligouridylation is a very fast, at the moment at least, it's believed to be a very fast process. So you will not find many of oligouridylated microRNAs in your samples unless you're specifically looking for them.
But thinking of the question before, if I have active microRNAs, and I can synthesize them with any synthesizer, nucleic acid synthesizer, and I uridylate it, then I have a stable microRNA.
No, if you uridylate, you've got a degradation, faster degradation. It's the other way around.
Okay. So, questions? No questions?
Is that a question up there, or are you just standing? No.
Okay.
Oh.
So,
Okay.
You said that you get about 1.5 cycle shift between your FFPE and cryo. So this is more or less a 3-fold expression difference.
Right.
And you also showed that some genes are only present, especially the low copy genes, if you have 50 or 100 cells.
Yes.
But for the low copy genes, this 3-fold expression difference might be the essential difference to detect them, or?
That is true, but the effect of microRNAs in general on translation is just a very tiny bit. And in one cell, in order to cover all the target sites of a microRNA, you need a very high expression to get the biological effect. So most of the time you need a very high microRNA expression to get translational inhibition in a significant amount. So the microRNAs that you sort of push over the edge by introducing the formalin fixation are probably not biologically relevant.
Okay. So thank you.